Hi, Mike. How are you?
Hello. It was a very long trek. I drove all the way to Mexico today.
Okay. Well, it is a real pleasure for me to introduce our speaker for the plenary lecture. Mike Blatt was educated in US and Canada, obtaining a dual baccalaureate degree with honors from the University of Wisconsin in Madison, and his PhD from Stanford University, and he held a postdoctoral fellowship at Yale University Medical School with a National Research Service Award before moving to the University of Cambridge in UK. Mike was a lecturer, reader, and held a personal chair at the University of London, White College, and Imperial College in London. He took up the Regius Chair of Botany at Glasgow University, where he heads the plant science group. He has held a number of advisory roles and serves within the UK and internationally. He is the editor-in-chief of one of the most highly cited international journals in plants, Plant Physiology. His research line is on ion channel structure and function in plant cell signaling, mechanisms and roles of membrane trafficking proteins in cellular homeostasis and stress responses of plants. But most of all, Mike is a great person who hosted me several years ago during a sabbatical stay in Glasgow. Next year, July 1st, will be the 65th anniversary of the Mexican Society of Biochemistry, and we are preparing several academic events, beginning with his talk here today. How do you get Stomata to shut up, or at least to talk in turn? Mike, many, many thanks. I know you just flew from Glasgow to London to make time in your so busy agenda. Many, many thanks. At least I can see you, at least, in the screen.
Yeah, well, can you send some tequila over while we're at it?
Oh, yeah, I can send you a bottle. Don't worry.
A virtual tequila. Now, with all those accolades and so forth, I'm not sure I know how to drive this thing, so we'll have a go. So I have to open the share tray, is that correct?
That's correct.
Yeah, okay, and then let's just see if I can get this to work. So you can see my screen now?
Yes, we can see your screen. Very nice.
Okay, so here we go. Let's just see if I can get it to work. Ah, there we go. So you can see that. Fantastic. Okay,
yes, so we can see that.
Well, this is going to be a slightly different talk than I suspect that would be appropriate for a biochemical society, but I think there will be a little bit of biochemistry in here. But I'm going to focus much more on actually how you get this. How do I get that thing out of there? Let me just figure out how to make this smaller. Can I? Oh, there's my mouse. Can I get it out of the way? Yes, there we go. Okay, right. So that is now out of the way. So I'm going to focus on Stomata today, and my apologies to Winslow Briggs, because this really was his title a few years back, not mine, but I loved the title and it's brilliant. So it's really about how do you get Stomata to speak in turn. And I'm going to begin with a little bit of background. So what you're seeing here is actually 10 years from almost 10 years, it's 8 years, 2005 through 2013. Each image is 1 month, and the coloration, the scale you'll see on the lower left, shows you the amount of water in the first kilometer of the atmosphere. So blue is very strong or high level of atmospheric humidity, white is essentially none. And you'll notice the fluctuations season by season, so the humidity shifts between the northern and southern hemisphere, as you would expect. And what's interesting is you see some areas which are perhaps lower in humidity or higher than you might anticipate. So a lot of Australia, for example, actually has fairly humid atmosphere, much more than I would have anticipated. And the west coast of South America, the Atacama and so forth, is unbelievably dry, but we know that already. Right, so this is really what it's all about. And this is where the big challenge is going to come. We already know this. These are some images that were taken, the fires in Australia back in 2017, drought in the Western United States in 2018, and the scale, or the chimograph that you see on the right, shows the relative temperature normalized to basically the period from 1955 through about 1980. That's a year-by-year account for the average temperature. It's actually month by month as we go along over that time. No, it must be year by year, sorry. It goes from 1901 through 2016, and it's by continent and then by country. So A to Z, going down for the Americas, then A to Z for Europe, A to Z for Africa, and so forth. And it's very obvious that the temperature is rising rather steeply, and has been since the early or mid 1990s. And this is clearly a problem that, as I'm sure you're aware, has been the focus of a lot of the discussion around the meeting in Glasgow just a couple of weeks ago, and is something that we are all going to have to live with in the coming years. Stomata play an important factor in this because they're very important for water use. And in fact, when one looks at the amount of water used on the planet, 70% of it goes into agriculture. And I always present this to students in undergraduate lectures. I point out to them that the global population has at this point more than doubled since the late 1960s. But in that same time period, our use of fresh water has increased sixfold. So agriculture is the main driver for water use on the planet, and its increased agricultural water use and withdrawal from water tables has increased quite dramatically, even in the UK, which you wouldn't normally think of as a drought or drought-ridden area. But parts of the UK actually have less waterfall than Northern Africa. Not Scotland, I have to admit. I put this slide in, again, to emphasize that factor, because this highlights the transition from rather poor weather prediction in the period up to the early 1990s and the subsequent transition to accurate weather prediction, so accurate that in most parts of the world now you can literally set your watch by the weather prediction. And it all comes down to a trial that NOAA started in the early 1990s, bringing in physiological use of water, basically evaporation or transpiration from croplands, to help calculate the distribution of water and weather patterns across the United States. And the image that you see there was the prediction for major floods down the Mississippi in the early part of July, which was predicted a full week in advance based on the water transpiration patterns and the predicted waterfall across the northern US, which is indicated by the gray and now dark black areas. So that was the first success of this approach, and it's now a standard approach, but it emphasizes the point that stomata and transpiration plays a huge part in our weather patterns on the planet. So stomata are obviously an indirect modeling target, and these sorts of models are really important for making large-scale predictions about the distribution of water on the planet, photosynthesis activity on the planet, and so forth. But they're all based around linear circuit models. And as a consequence, while they have great application in sort of phenomenological prediction, they're pretty useless when it comes to understanding and improving or working with the canopy of crops in order to improve their activity. In other words, in particular, improve their water use efficiency. So the big problem here is these models don't make the essential link to the microscopic molecular mechanics that goes on in the plant that drives how stomata work. So what we would really like to be able to do is to understand that process. And I think I'm jumping a little bit ahead here. So basically, what does stomata do? They balance the demands for water and carbon. They take advantage of gas exchange and maintain a high hydrated environment within the leaf. So within the leaf's air spaces here, I hope you can see my mouse. Here's a pair of guard cells. There's a pore right there. And it's through this pore that both CO2 enters for photosynthesis within the leaf, and we lose water also via transpiration. And this is often quantified as water use efficiency, which is quite literally the amount of carbon fixed divided by the corresponding amount of water lost during that same period of time. There are many ways of measuring it, but just keep that measure in mind. And one of the big problems with any effort to improve water use efficiency is the consequence of affecting carbon fixation. So I'll just point you to one set of data. So this is a paper that appeared in Plant Physiology a few years back from Julie Gray's lab, who has been working on EPF factors which are involved in stomatal development. And they've been able to manipulate the density of stomata on the leaf surface. And the consequence of reducing the density of stomata, here you see some examples. So that's basically the density. These are various EPF mutants. And with more severe mutants, you can seriously reduce the density of stomata. And when you do this with some of these mutants, so one of the intermediate ones here, you get a significant increase in water use efficiency that you see here. So WUI, basically the amount of carbon per amount of water lost. But you do so at a loss of carbon gain, usually not huge, so on the order of 5 or 10%. But when one thinks that about the implications for grain yield, that becomes quite important. So one of the questions that we can ask is, how can we go about improving water use efficiency without the implicit trade-off in carbon fixation? So ideally, what we'd like to be able to do is to take our knowledge of what's going on in the canopy and work backwards to the components in the guard cell and in the stomata that determine how the stomata work. And in order to do so, we first have to construct mechanistic models that define the characteristics of all the enzymatic and transport reactions that go on in the guard cell and at the guard cell plasma membrane particularly, translate those into what goes on at the cellular level, then at the leaf, and then ultimately to the canopy. So we'll work forward with the idea that once we have developed that scheme, we can then work backwards for an understanding of how best to improve water use efficiency and crop efficiency. Okay, so just a brief summary. There have been a lot of people who've worked on this program, and I've just highlighted with the asterisk some of the people who have worked particularly on the modeling side over the past decade or so. This is a project that has been going on in my lab for 15 years now. Adrian Hills is a fantastic computer programmer who has worked with me now for a quarter of a century. I have two superb electrophysiologists currently in the lab, Hao Nguyen and Cécile Leffelon. I also have a number of colleagues who are working on the molecular side as well. And there have been a number of colleagues, including Alex Agramov, Jean-Hua Chen, Izhou Wang, Maria Papanazzo, and Marika Jezek, who've all gone through the lab and contributed to the modeling effort. And of course, I also have a number of colleagues who've collaborated over the years, in particular, Ari Lev, via Lev here, who has been instrumental in helping to design and develop the modeling, Simon Rogers, Tracy Lawson, Howard Griffiths, and also other colleagues, also not listed here, who have contributed as well. So let's just begin with the idea that we can start with guard cells as a model system. And one of the big advantages of guard cells, of course, is they're isolated. They have no plasma desmata when they're mature, so they're essentially a semi-closed system. The next thing is that we know a lot about all the transporters, pumps, carriers, channels, and there's quantitative data available for all of the major ions and their buffering characteristics. Furthermore, we know a great deal about the physicochemical constraints, the conservation of mass, charge, osmolality, and we can define each of the various transporters with very accurate flux equations. So it's not beyond the wit of man to construct mechanistic models that take into account all of these properties at the cellular and subcellular levels. However, it's a very complex system. So if you look at what goes on in the guard cell, understanding it is not quite so straightforward. So as the stomata opens and closes during the day, there are some rather surprising changes in all of the ion contents. So for example, calcium goes up during the day, comes down during the night. Chloride goes up but drops during the day, whereas it's replaced often by malate. Potassium goes up and then comes back down, and so forth. So how do we put all of that together? Well, let's start with the individual building blocks. So this is where Xianghua Chen came in, because I set him the task right from the start to construct a detailed database of all the information that we have for all of the transporters and metabolism in the guard cells. And we drew from a huge body of data. I don't know, these fan-fold papers that old printers used to use. He ended up printing it out on these sheets, and the fan-fold, and he had 4 sheets, one for ion channels and one for pumps for each of the major membranes. So 4 sheets, plasma membrane and tonoplast, and each of these sheets was the better part of 8 feet long. Huge, long lists of each of the characteristics. But that was a wealth of data that we could then put together to understand how the whole process works. And we ended up ultimately with 2 primary publications in 2012, both in plant physiology, and a third one which followed shortly thereafter, which demonstrated the utility of the model. So what you see here is the first generation windows, which allows us to have a view of the current voltage relations of the plasma membrane and of the tonoplast. Don't worry too much about the IV curves here. I will explain a little bit, but you can think of these as essentially a measure of enzymatic activity in much the same way as a Michaelis-Menten plot is. So it's a plot of the capacity as a function of voltage as it were, a virtual substrate. It also provides us with a window for fluxes and also a chart recorder. So we can see how things evolve in real time using this modeling scheme. So what's built into it? Let's just have a quick look, and then we'll look at what we have been able to do with it. So first of all, pumps and ion-driven carriers are described with a Slaiman-Finkelstein type reaction kinetic equation, and that's just illustrated here. One doesn't need to worry too much about the details, but the important thing is here that we bring in voltage in this enzymatic carrier cycle as a driver between the 2 states across the membrane. We can bring in substrates as direct reaction kinetic factors, as illustrated here. And we can also bring in ligand controls, as many as you might wish to have for each of the different pumps or carriers. For ion channels, we follow very much the same sort of idea, but here we make use of the Goldman-Hodgkin-Katz flux equation, and then build into that the voltage dependence of the open probability. So we can put in gating characteristics associated with membrane voltage, and we can build in ligand control, such as control of ion channels by cytosolic-free calcium. And finally, we have to deal with metabolism, primarily that of malate, because malate is one of the major osmotica, the main organic osmotica in guard cells. And for this purpose, we can simplify the enzymatic processes down to a very small number of, as it were, how shall we put it? So we subsume all of metabolism into a very small number of steps. So we can go from carbon dioxide to sucrose. Sucrose then equilibrates with malic acid, and we also have a sink or consumption of sucrose through metabolism itself. And lastly, we need to draw, sorry, I'm jumping ahead of myself. Constraints, really important, is electroneutrality. There can be no net charge buildup across the membrane over any significant period of time. So charge, by basic laws of physics, must balance over in the steady state. In other words, any current carried across the membrane in one direction, for example, by a proton ATPase, has to be balanced by current returning through other transporters back into the cell. And lastly, we want to drive the system, and we've made some very simple, straightforward assumptions that are well supported in the literature, basically that the primary ATP-dependent pumps rely on ATP produced through photosynthesis, and therefore we can link the activity of those pumps to light. And then all we do is we drive the entire system through a diurnal cycle of 24 hours with some sort of light during the day and dark during the night. And we allow for the activity of the pumps to drop to some basal level in the dark. So here we just have a very simple sawtooth. We can bring in sigmoids, or you can actually, in the new version of the model, describe your light program as you wish. But for our purposes, we'll just use a relatively simple light system. Okay, so all of this does one thing. It produces content, solute content. So that's basically what you have here, total content, which sums up in the vacuole and the cytoplasm. We assume, as there's plenty of evidence, that there's no turgor across the tonoplast, so that must equilibrate osmotically across the two. So we can equate the content and the volume, as indicated here, and we then can bring in the Van Hoff relationship to describe the total volume in relation to the total content, the apparent isotonic content outside, and the pressure-volume relationship. And we can later on, I'll mention this as we go on later on, we link this to the water content in the air. So we're basically, this is an engine for producing solute content in silico, and then all we do is relate the solute content based on established empirical data, linking that to turgor pressure and stomatal aperture. And we can do that for any species for which there's sufficient data to construct this kind of a model. So here's some preliminary data. These are some of the very early outputs that we published in those first few papers, and it just shows the variation in membrane voltage with day-night cycles, 12 hours light, 12 hours dark. So this is 5 days here. This is the aperture, turgor, volume, and aperture. This is the vacuolar volume percent, so the percentage of the volume taken up by the vacuole during the day. And this also varies as you would expect. So as the cells swell up, more of the cell volume is taken up by the vacuole. And we can then look at how aperture, volume, and turgor relate to external parameters. So for example, pH, external calcium, and external potassium chloride. So these are very early data. It shows very nicely that as you drop pH in the model, as you would expect in vivo, the aperture increases. As you increase calcium, you see a very steep decline in the aperture in the open state. There's no effect in the closed state, of course, and that's also shown by the modeling. And likewise, in the open state here, the aperture increases with increasing potassium chloride up to about 20 or 30 millimolar, and at that point it more or less levels off. All of that is, there are literally hundreds of papers that demonstrate precisely this. Okay, so all well and good, we have a model which can do something and it more or less reproduces what we know. but it also tells us a lot of other things. And I'll just give you a few tasters here. So this is, again, that same cycle, 5 days. Here's the potassium going up in the daylight, in the cytosol, in the vacuole, in the cytosol, and where's my mouse? There it is. And here in the vacuole. And this is the flux in terms of the directional flow across each of the membranes. Now, bear in mind, we're activating the proton pumps at the plasma membrane, and of course also that we have a proton pump at the tonoplast. So what happens? Well, you activate the pumps, you get an increase in potassium uptake because you're energizing the membrane, and you see an influx. So here negative means an inward flux of potassium, the chemical compound, not the charge, okay? So here we have an influx of, mouse, come on, wake up, there we go, influx of potassium. And you'll notice that at the same time, you have an efflux of potassium across the tonoplast. And then at the end of the day, it reverses. You get this massive dump of potassium across the plasma membrane, outwards, and an influx of potassium across the tonoplast from the vacuole into the cytosol. So these two membranes, even though they are not connected together, somehow talk to each other in such a way that we have a net flow of potassium across the plasma membrane and tonoplast into the vacuole during the day, and the whole thing reverses at the beginning of the night period as the stomata close. There's nothing intrinsically built into this to say, hey, plasma membrane, you need to be talking to the vacuole and vice versa. It just does it. Okay, here's an even more stunning example. So here we're looking at malate flux. So that's the malate metabolism and sucrose up there. But let's just focus here. So malate goes up in the vacuole very steeply during the day, drops during the night. It goes up a little bit in the cytosol and drops essentially to zero during the nighttime. And let's just look at the fluxes. So these are the total fluxes. These are, again, broken down in the plasma membrane and in the tonoplast, but let's just look at the total flux. So during the daytime, as illustrated here, there's a net efflux out of the cytosol and into the vacuole, but there's absolutely no flux across the plasma membrane. Of course, malate is synthesized in the plasma membrane, in the cytosol, so you would have thought, well, okay, it should also flow across the plasma membrane. It doesn't. Then at the end of the day, all of a sudden we have this massive dump of malate across the plasma membrane out into the apoplast, and a parallel corresponding influx across the tonoplast from the vacuole into the cytosol. So somehow this system is able to know, if you want, in inverted commas, know that it should move the malate into the vacuole during the daytime, and then at the end of the day, it should move all of that accumulated malate, or most of it, out of the vacuole into the cytosol and across the plasma membrane into the apoplast. Nothing ad hoc built into this to say that's what it should do, but these two membranes communicate. Okay, so let me give you a case study. This is one case study of a number that we've now carried out, and this is based on work that was published in 2008 describing the SLAC1 channel. And what was observed in those early publications was that if you eliminate SLAC1, which is the anion channel at the plasma membrane, then you see that the, come on mouse, where are you? There you go. Then the stomata close relatively slowly. And you also see that there's a buildup of, let's just focus on the red bars here. There's a buildup of malate, and there's a buildup of a secondary organic anion fumarate. There's an accumulation of chloride, but interestingly enough, there's also an accumulation of potassium. Well, you'd say, okay, you have to balance charge, fair enough. But how do you explain this? Although this is a pathway for chloride efflux, that's well documented. So that would explain why you have a much slower stomatal closure. How do you explain that the stomata open more slowly in these mutants as well? Okay, so we put our problem to the model and asked the question, can we reproduce the characteristics of SLAC1 in this model, what we call on guard. Okay, so here are the data. So here's the aperture, and just to walk you through this, this is the wild type open, closed and open, and then the mutant closed and open for each of these different parameters. And you can see that the first thing is the stomata are much more open, whether closed or open, and the dynamic range is much smaller. You'll also see that the amount of potassium that's accumulated is higher, both in the cytosol and also in the vacuole. Oops, sorry. Likewise for chloride and likewise for malate. So we've reproduced the experimental data, at least qualitatively. We can also reproduce the opening and closing rates. So here's the wild type. That's the opening rate in microns per hour. And you'll notice that the SLAC1 mutant is much slower by about a factor of 2, or 50% slower if you want. And we can also look at the closing half-time. Here's the wild type. The half-time is pretty quick, but in the mutant, it's about 4 times slower. Okay, so what's going on here? How do we explain the slower opening rate as well as the slower closing rate? Well, the model suggests that the main reason for that is a substantial reduction in the activity of the inward potassium current. And now you have to get your head around a little bit around these IV curves. So this is a measure of the activity of the channel as a function of membrane voltage. The negative current simply is convention that implies movement of positive charge into the cell. So this is the inward potassium current. So it allows potassium into the cell. And as you drive the membrane to more negative voltages, such as if you were to activate a proton pump, then you see an increase in current. And you'll note, so this is essentially the capacity for current carrying through the channels as a function of voltage. And for the SLAC1 mutant, we see about a 80% reduction. Interestingly enough, for the outward potassium channels, which mediate potassium efflux during stomatal closure, we see about a 2-fold increase in the activity in the mutant. And what Izhou Wang was able to demonstrate is that's exactly what you see in the cell. So here are the actual experimental data. There's about a 70 or 80% reduction in the inward K channels and about a 2-fold increase, a bit less perhaps, in the outward K current. Okay, how do we explain that? pH and calcium. So the model predicts that the pH in the SLAC1 mutant, so again, it's wild type, closed and open, SLAC1 closed and open. The pH in the SLAC1 mutant is substantially higher, whether it's closed or open compared to the wild type. That's in the cytosol. In the vacuole, the pH is pretty much smack on where you would expect it with malate buffering. And if you look at the calcium levels, and this is where it really gets interesting, closed and open for the wild type, you have that increase in calcium. But here, in the closed state, the calcium levels are about the same as in the wild type. But when you have the open state, the calcium is substantially higher compared to the wild type. And that's pretty much what we find in vivo. So first of all, we see a hyperpolarization of the membrane, and that's borne out experimentally. So these are just measurements at 3 different K concentrations. And this is the predicted behavior for the membrane voltage at the plasma membrane for the wild type, and here for the SLAC1 mutant. And this is the calcium measurement. So here is the predicted calcium behavior for the wild type, and here for the mutant. So this is the free calcium. But let's not worry about all the rest of the gubbins down here. That's just the components. But the important thing is the free calcium is substantially elevated much of the time in the SLAC1 mutant, and that is borne out in experiments here. So wild type, SLAC1, and this is a complemented line to show that the calcium levels are back more or less to normal. Likewise, pH, we anticipated the pH would be substantially higher much of the time in the SLAC1 mutant, particularly in the cytosol, and that is exactly what we see. So these are the measurements, wild type, That's the SLAC1 mutant. In fact, it's substantially higher than we had anticipated. And we can even calculate the buffer capacity by weak acid loading. And there's about a 3-fold increase in buffer capacity, which was consistent with the increase in malate content. Okay, I'm not going to bore you with the calcium measurements now, but I just want to summarize what how we interpret these data. So in the wild type, we would normally have this SLAC1 channel, which allows some leakage of chloride and malate out, even in the open state. We have some membrane voltage. We have a proton chloride symport. We have a calcium channel, and we have the, just for argument's sake, just show the inward going K channel. In the SLAC1 mutant, we lose the channel that mediates chloride and malate efflux, and the consequence of that is we lose the pathway, the leakage pathway that allows negative charge out. So we get a hyperpolarization of the membrane. A hyperpolarization of the membrane drives an increase in calcium influx. We get an increase in free calcium as a result, and we know that free calcium inhibits the inward K current. As we have a buildup of malate and chloride inside the cell, two other things happen. First of all, we have chloride acting as a inhibitor of the chloride proton symport. So that reduces the proton load on the cytosol mediated through this transporter. And more importantly, the buildup of malate acts as a inhibitor of malic acid synthesis. And again, that reduces the proton load. So we see a reduction in the protons or an increase in pH, and likewise, that's well known to affect the activity of the potassium channels. And while I haven't shown you the data, we can also show that by buffering both pH and calcium, we can recover or restore normal behavior, at least of the potassium channels at the plasma membrane. So that fits beautifully. We can use the model to understand the connections through metabolism, potassium metabolism with calcium and pH, how that affects, how the SLAC1 mutant affects the activity of the potassium channels. So now we need to bring in water and CO2, and of course this brings us to the point where we need to extrapolate the model to, or this modeling from the single cell level or from the stomata level in an epidermal peel, to the point where we can understand what goes on in the whole plant, or at least in the leaf. And obviously that means we need to bring in the exchange of water vapor inside the leaf with water in the atmosphere. So we've taken a leaf out of the page of Peake and Mott, who suggested back in 2011 that there might be a site with which the guard cells equilibrate in the epidermis. So here are the guard cells, equilibrate behind the epidermis, I should say. So here are the guard cells, here's the epidermis, here's the mesophyll, there's the xylem, and we know that inside the leaf, in the leaf airspace, you have a hydrated environment which is pretty close to 100% humidity. But this allows, with the pore open, there is some leakage of that hydrated environment out of the pore, leakage of water across the pore. And so we can imagine that there's likely to be a gradient of water vapor pressure or partial pressure of water between the inside of the leaf and the outside. And the main point that Peake and Mott pointed out was that you need only have a very small decrease in the relative partial pressures of water close to the guard cells, which equilibrated with the guard cells in order to effectively increase the osmotic strength or the osmotic load, the draw on the cells. And that draw, which would pull water out of the cells, would cause the stomata to close. So you could have a relationship simply by the physical mechanics of water equilibration with the water vapor just behind the stomatal pore. Now, in their model, in their description, they allow that equilibration with the cytosol and the internal water content of the guard cell. But we know, of course, that that can't be the case because there has to be equilibration with water in the cell wall. So we brought that in, and as a consequence, we can now align that kind of a model with the more typical model of water flow along the surface of the cells, and still allow for the relationship of the apparent osmotic content outside with the ratio of the saturated water vapor with the water vapor content just behind the pore at this P site, as they called it. It turns out that has a huge effect. So even a 0.6% decrease in the relative water content just behind the pore is enough to increase the apparent osmotic load in the cell wall of the guard cells by the equivalent of 300 milliosmoles. So if we do that, what do we get? Well, by bringing in that, allowing water to equilibrate through aquaporins, because we also need to bring in aquaporins, and then allowing for equilibration of solutes according to the osmotic gradients as well, we reproduce very nicely the behavior of the stomata in the whole plant. So here we have relative water feed. I can explain that, but let's just assume for the moment that it's equivalent to the relative water content. It's not too far off. So the relative water feed with essentially 100%, or 100, I should say, rather. And here we have a step in relative humidity going from 85 to 40% and then back to 85. And when the plants are well watered, so RWF of 100, essentially they don't pay any attention at all to the change in the atmospheric water content. But as you start putting pressure on the plant, so for example here with a relative water feed of 10, now you see a very substantial drop in stomatal conductance as you drop the relative humidity outside, and then it recovers when you bring the humidity back up. And that's exactly what you see in real life. So these are measurements from Tracy Lawson's lab, from Sylvain Vialet-Chabron. What's also nice is that we see in the model that this corresponds with the change in osmolarity, or osmolality, which is exactly what you would predict if you were drawing water out of the cell. And here we have measurements. So the surface here, water feed, the relative water pressure, or water component in the atmosphere, and the stomatal conductance. The data points are actual experimental data, some of which you see up there, and the surface plot is the plot that comes off the model. One of the neat predictions of this is an observation that I made years and years ago when I was a postdoc in Ina McRobbie's lab in Cambridge, when I had done some osmotic experiments and discovered that as I washed out osmotica, I saw this massive change in the activity of potassium channels. And I never could understand it. But when we came back to this problem, it suddenly became obvious. So what happens when you change this relative humidity outside? Well, one of the things that you see is you see a change in potassium, malate, and chloride, as you would expect. But you also get a small increase in calcium as you drop the humidity, mainly because of the draw of water out of the cells. But when you return the relative humidity back to 85%, you get this huge undershoot in free calcium inside the cell, which then recovers. And that correlates perfectly with the overshoot in the activity of the inward K channel. So here we have the modeled output for the outward K current, and here for the inward K channels. Here's the control. You reduce the relative humidity, you see a decrease in activity in the K channel current. And now you return to 80% relative humidity, and you see that there's, at least for a period of time, a substantial overshoot in the activity. And we know that these channels are calcium sensitive, and that's exactly what you see experimentally here. So again, the model allows us to make some really important predictions about what goes on and the connections between water vapor within the leaf and how the stomata behave and respond to that, and what the components are that drive that process. Now, let's finish up the story. I'm going to try to do this relatively quickly because I know that we've got only about another 10 minutes, and I want to leave some time for questions. We need to bring in CO2. And it turns out this is remarkably easy because all we have to do is to work the water problem backwards, because the diffusion pathway for CO2 is essentially the same as it is for water. Now, why have I done this? All right, so this is what I wanted to get to. So we can define a diffusional pathway through the pore and the leaf. Most of the resistance occurs across the pore, and relatively little, a smaller percentage within the leaf to the mesophyll. And we then simply bring in the empirical relationships for assimilation of carbon. So basically we say that carbon has a sink in the mesophyll and has a source in the atmosphere, which is exactly the opposite of what goes on otherwise for water, where the source is in the leaf, the surface of all the mesophyll cells, which evaporate water, and then the water diffuses out the pore. So the problem is not to describe the gradient of CO2, but rather to ask the question, what is it that we need to couple the changes in internal CO2 in the leaf, PCI, what is it that we need to couple PCI to in order to get the stomata to behave? So essentially we can construct hypothetical models, so these sort of thought experiments where each construct is essentially a description of a hypothesis, which we can then test according to what we know about how stomata behave. And if you're interested, this is now in Jezek et al. 2021 in Nature Plants, volume 7, 1301. So here we have the model. So this is what I showed you before for the water, and here we have it for CO2, but now CO2 is diffusing from the atmosphere through the pore. Again, we have this P site, which defines the CO2 content along that diffusional pathway, just behind the guard cells. And it then goes to the sink in the mesophyll, where it's consumed and fixed into sugars. So here we have the model output. And I'm not going to tell you how we get there, but I will do in a minute. And here we have the experimental data. And you can see that there's a pretty damn good match between the two. When we find the right targets for CO2 to act in the guard cells, we get exactly what we see in vivo. So these are measurements in 50, 100, and 200 micromoles per meter squared of photosynthetically active light. And this is the calculated PCI inside the leaf, and this is the modeled PCI within the leaf. And again, they're very close. Here we have 50 micromole par, and we're stepping down in CO2. Here we're stepping up from 400 to 1,000. Here we're down from 400 to 100. Experimental data, again shown with the data points, and the surface plot is the output of the model. So really nice fit. So how do we get there? Well, it turns out, you might say, oh, well, CO2 must be acting on slack. It doesn't. If you target CO2 to the slack channels, or for that matter, to the ALMT channels, which also carry anions, in this case, the malate anion, you have absolutely no effect. If you add in the endomembrane calcium channels as a second target for CO2, or for bicarbonate, it doesn't matter which, we can use both. Again, there's essentially no effect of changing CO2 in the atmosphere. In order to get a response, you have to have CO2 target the endomembrane or vacuolar calcium ATPases. And if you add in SLAC and ALMT as targets for CO2, it makes no difference whatsoever. In order to get the best response, we have to target both the calcium ATPases and the calcium release channel, V calcium in, at the tonoplast or at the endomembrane, in order to get the system to give essentially physiological kinetics. And that's all you need. You don't need to do anything else. So we can define the absolute minimum targets for this kind of behavior. It doesn't mean that CO2 doesn't target other channels like SLAC or whatever, but it says that simply targeting those is not sufficient. It's not necessary either. Okay, so we can go through essentially the same kind of tests. What do we see? One of the predictions is an overshoot. Again, as in the case of relative humidity, if you raise CO2, you inhibit the inward K current, you activate the outward K current. If you drop the CO2, you get an overshoot in the activity. Again, this is linked to changes in cytosolic free calcium. And one of the things we noted was that there's also an undershoot in that calcium. So as you return the CO2 back from 1,000 ppm or 1,000 microbar down to 400 microbar, then you get this undershoot and then a recovery. So we thought, maybe what's going on here is that we're depleting and recovering calcium into endomembrane stores. So what would happen if we altered the CO2 levels or light levels, doesn't matter which, let's just focus on CO2. What would happen if we alter the CO2 levels frequently, raising them and lowering them, raising them and lowering? And what you find is in wild type, as long as the period in between the pulses is relatively long, so 45 minutes or an hour, you reproducibly get this rise and undershoot. But if you shorten that period, you start to see a latency. And that's what this set of curves, this is very complicated, I admit, but let me walk you through it. So here we have the latency. So there's a first pulse in CO2, and then we have a period of 15, 30, 45, 60, or 120 minutes before the second step in CO2. And what you see is that the rate of decline with very short intervals is quite slow, and it only recovers after about 45 to 60 minutes. And that's what's plotted here. So this is the rate of the decline with raised CO2, and these are the experimental data there and here. So there's a pretty good match between the modeled prediction of a latency and the experimental data. And we thought, now this is really neat, because what that is telling us is that the response to fluctuating environmental conditions is linked to the calcium stores and recovery of those calcium stores following stomatal closure. So what would happen if we manipulate the ability of the cells to recover the calcium stores? And the obvious thing to do is to mutate the calcium ATPases. So we turned to Jeff Harper. Jeff had already a number of double and triple mutants, both at the endomembrane of the ER and also of the tonoplast. And we tried both, and they both work equally well. And in the model, in fact, all you need to do is knock out about 50% of the pumps in order to get the kind of behaviors we see here. So here the wild type, this is with a 45-minute delay between the different steps. And you can see that the decline in each of these cases in the stomatal conductance is pretty much the same in the first, second, and third steps. But for the mutant here, the ACA411, which is the tonoplast mutant, double mutant, here's the first step. There's the second step in CO2, and you can see now it's quite a bit slower. And here's the third step. It's even slower still. And that's just a plot of the rate as a function of the first, second, and third step. And indeed, the model, as well as the mutants, show exactly that sort of behavior. And all of this is linked to the ability of the cells to respond the second and third times to a step in CO2 in their ability to raise cytosolic free calcium. So we think what's going on here is a pretty simple story. CO2 affects PCI in the leaf. PCI affects the calcium ATPases and the calcium release, and you then have to recover calcium from the cytosol into the calcium stores before the cycle can operate again. So if you challenge with fairly high frequency fluctuations in light or CO2, then in the mutant, what happens is that there's not enough time to recover enough calcium in the stores, and the second and third cycles are very much reduced in their ability to raise cytosolic free calcium. So one might think, okay, well, obviously the way to go in terms of increasing stomatal behavior is to speed up calcium recovery. And that's a story for another time. But this has direct implications for how stomata behave in real life. And this is just an emphasis of that. I'm running out of time, so I'm going to stop at this point. I have more, but I don't want to belabor the point. In real life, in the field, plants experience fluctuating light. Well, okay, let's face it. In Glasgow, that's not the case. It's always dull and gray, okay? But in most parts of the world where you grow crops like wheat, rice, barley, etc., you have fluctuating light as clouds pass overhead. And the real problem here, and I'm going to go back now if I can. I don't know if I can. Come on. The real problem here, oops, there we go. That's what I wanted to get to. The real problem here is that during fluctuating light, there's a carbon deficit and a water excess. So the stomata respond relatively slowly compared to the response of photosynthesis, which is driven pretty much directly by light. So there's a carbon deficit associated with the ability of the stomata to reopen. And at low light, before the stomata close, there's a water loss excess for which there's no real carbon gain. And if one could speed that up, you could substantially improve water use efficiency without a loss in carbon gain. So I think that's the big message here. I'm trying to figure out where we are now. Bear with me. There we go. So that's what I wanted to end up with. So this is our model. Now we can go from what's going on at the molecular level in the different transporters to the whole ensemble of transport to what goes on in the cellular behavior in terms of stomatal aperture, solute content, membrane voltage, and so forth. And we can take that to what goes on in the leaf in terms of gas exchange, and now to water use and water use efficiency in the whole plant. And I'm going to leave it there because I think time has run out. Okay, now I've got to figure out how to get back to see you.
Many thanks, Mike. Is there any questions?
How do I get back to see you? Sorry.
You have in the same arrow, just put stop share screening.
Yeah, I've got to figure out how to get to stop share screening. There we go. Stop presenting. Fantastic. Got it.
Okay. Toon, please.
Yeah, Mike, very nice story. Thank you. I have actually two questions.
Well, thank you too. Hello. And we should have been meeting in Mexico, man.
Exactly, yeah, for sure. Hey, Mike, I have a question on the malate.
Yeah.
So I was wondering if you have malate transported to the outside, to the apoplast, where it's quite acidic, or into the vacuole, which is quite acidic, that as a weak acid, actually the malate would be able to diffuse across the membrane without any transporters. So do you see this, or is this measured, or is this impossible, what I'm saying?
Well, the malate has to transport across all evidence says the malate transports across as the M2 minus form. So malate itself, MH2, may well diffuse, but it appears to be relatively slow, as far as I know. There was some work from Kaiser years and years ago, which suggested that it might contribute a small component to the to changes in malate, but it's relatively small. And we know that there are ion channels that play parts in transport of malate. And if you eliminate those, you see a big effect in terms of malate transport. That's from Enrico Martinoi's data. So I think the answer is Yes, it probably can, but the rates are fairly low, and the transport is primarily through ion channels.
Yeah. My second question is about your model, where the post-translational modification comes in. I mean,
good question.
In the end, it's more than only light and CO2. There is also changes in temperature and.
Of course it is. Absolutely. So, I mean, one of the big limitations at the moment of the model as it stands, and you're quite right here, is that we have not built in explicitly any way to deal with post-translational modification. In fact, it is already there in the sense that if post-translational modification does nothing more than modify the number of active pumps, channels, or whatever, you can simply manually change that. So you can say, I'm going to change the number of potassium channels from, let's say, 300 GORC type potassium channels, and they're going to be phosphorylated, and that activates them. So all I have to do is increase the effective number from, let's say, 300 to 500. So that would be the equivalent of a post-translational modification numerically in the model. Now, what we're about to do, and once we have actually at the moment we're working on a quote eco-friendly model or eco-phys friendly, so that people don't have to know how to read IV curves in order to make use of the model. And I am hoping that will be out sometime in the early spring or at least early summer of this coming year. But once that is done, the next big challenge, and it's actually not a challenge, it's just a task, is to build into the model the ability to change the numbers of any transport or metabolism in a diurnal cycle. And the main reason that I'm interested in that is I want to see what is the minimum that we need to build into the model in order to convert the model as it stands now into a CAM plant. Because we all know that CAM plants, the stomata of CAM plants work backwards. So they close during the day and they open during the night. And there's a lot of data that says, primarily from John, go and help, and at Nevada, and James Hartwell, who've been working on this now for a number of years. And they've been able to show that there are fluctuations in several of the major ion transporters, in particular the SLAC channel, the ALMT channel, homolog in Kalanchoe, likewise the proton pump, at least one or two of the potassium channels. So there's already experimental data out there for the diurnal cycles of expression of these channels. And we can ask the question, if we vary the number of channels in some sort of diurnal pattern that mimics those patterns that have been shown experimentally, is that enough?
Mm-hmm. Well, I look forward to the other model also for our students, Mike. Thanks.
Yeah, okay.
Mike, so what was your conclusion? Did Stomata took you back?
I'm sorry, start again, Taya?
Yes, what was your conclusion? Did Stomata talk to you back?
Oh, well, I don't know that they talk back, but we are getting them to talk in turn. We're getting them to talk efficiently with photosynthesis, and I think that's the key point here. I didn't show the data for work that I did with John Christie 3 or 4 years ago, which we published in 2019, showing that you can express a synthetic ion channel in the guard cells that responds to light, and by turning that channel on and off with light, you can accelerate the stomata, and by accelerating the stomatal kinetics, you do actually get gains in water use efficiency and also gains in carbon assimilation. I mean, we were expecting, we'd be really happy with gains in water use efficiency, but in fact, we get both. So we see both a gain in water use efficiency and also carbon assimilation. So the big challenge in my mind now is, okay, we can do it synthetically. What do we need to do to make it work using the native ion channels? And that, again, is where the model is going to come in.
And how this can be applied eventually, you know, to start all these change in high temperature.
Well, I mean, you ask the question, and we will have a go at trying to answer it. But we now have the system working where we can work backwards. We can ask the question, what do we need to do, for example, to get the stomata to move more quickly, to talk in turn? And if we can do that, then we're already effectively doing what I was aiming to do. That is to work from the molecule up to the crop and ask questions about what is it we need to do at the molecular level in order to get the crop to behave the way we want it to.
Yes. Agustin, do you have a question?
Yes. Well, actually, I work with calcium, but not in plants, so I don't know a lot of the situations here. So from what I understood is that during the day, there is a new steady state, no? So you have the elevated calcium concentration. But I'm not quite sure exactly what is driving this new steady state. I mean, is the activity of the calcium channels at the plasma membrane or the endomembrane?
Yep, well, that's a good question. In a sort of a nutshell, I mean, I didn't show any of that information, but that was one of the very first things that excited me and got me talking with Alex Webb, who is also a calcium expert. So he had shown that, in fact, calcium does go up during the day. And that seemed counterintuitive because during the daytime, the calcium ATPases are activated because you have an increase in ATP, and you would expect that calcium would be sequestered more effectively or removed from the cell. Well, 95% gets sequestered. So most of it would be pumped into the ER or into the vacuole or whatever the case may be. And in fact, the model predicted exactly the opposite. And the explanation for that is that as you activate the proton ATPase, what you're doing is you're hyperpolarizing the plasma membrane quite substantially. And that hyperpolarization pushes the membrane voltage out of the range in which the ATPase is active and into the voltage range in which the calcium channels are active. So you're now promoting calcium entry across the plasma membrane. The calcium ATPases in the endomembranes compensate for that by accumulating calcium and storing them. But there's a shift in the balance, so you have a rise in cytosolic free calcium because of the imbalance and fluxes between the two membranes. So that's why you get this rise in cytosolic free calcium during the daytime. You also see a rise in the calcium stores, of course.
Okay, and in this case, the calcium sensitive potassium channels, do they have inactivation?
Yes, they do. So I mean, one of those really neat things about the OnGuard platform that I've been describing is that it produces data in real time. So it basically increments in very small increments in time and then allows the behavior of all the components to evolve with time. So you see the whole process as if it were in real time. Now, admittedly, you can run the model really quickly, so we can run through a 24-hour period in about 3 minutes. But you can also run it in a slower mode, and you can watch the whole process evolve, and it's fascinating. I've been accused of playing computer games, and that's really very close to the truth.
One, if I can, one last question, and it's not really about the model, but more like this idea that the Earth is getting drier just I mean, in the million years, not just nowadays, but from the course of the million of years. So it's I mean, what is driving this trend, and if you think there is an option to stop it?
I have a small visitor here, my grandson.
Hi.
Hello. Car.
It's actually
Oh.
Rafi's bedtime.
Oh, right.
So
I'll be done in a minute. Okay. Okay. I'll see you in a sec. Yeah. Well, again, it's an interesting problem because the issue is not so much the amount of water. I mean, Graham Farquhar, a good friend of mine in Australia, would say, well, yeah, water resources don't change because the total amount of water on the planet hasn't changed, but its distribution has changed. And that's certainly true. So, you folks are probably seeing a lot less water, and I have to say, we in the west of Scotland seem to be seeing an awful lot more than at this point. So, yeah, I think it's weather patterns. Obviously, the warming of the atmosphere. I'm not an expert in these issues, but clearly it's affecting the way the overall patterns of airflow and so forth are distributing the water around the planet.
Okay, thank you.
Certainly not in Scotland, Mike. As I remember, it rains every day, every single day.
Yeah, well, we put that on specially for you, Tara.
Thank you very much. Alberto, are you going to present the certificate? Yeah. Oh, it's for me.
Oh, fantastic.
Oh, I'm sorry.
Okay. This is a certificate to Dr.
Come on, Tara, you and I can exchange certificates.
Yeah, that's true. To Dr. Mike Blatt, in behalf of the Mexican Society of Biochemistry and the 8th Signal Transduction meeting, the organizers, Ulises Mesa, Ricardo Espinosa, and Alberto Olivares. Many thanks, Mike. You're probably getting ready for Thanksgiving with your family, so I really, really appreciate you took the time, and hopefully next time you will be here in Mexico.
I dearly hope so. I would love to come and visit you again, so I'm looking forward to that opportunity. But thank you ever so much for giving me the opportunity to join you on this meeting, because it's I sat in on a couple of the sessions. I really enjoyed listening.
Thank you very much.
Okay, take care.
Thank you.
Okay,
bye. Ciao, ciao. Thank you.